Good evening. I'm Pete Swenson, and it's my honor to welcome you to the fourth installment of the college's Curiosity Drives Progress lecture series. In each of these lectures, we feature 3 faculty members presenting about their work related to a larger theme, and today that theme is sustainability and the environment. Through this lecture series, we are showcasing the breadth of amazing research and teaching being done across the college by emerging new talents and seasoned experts in their fields. So my personal connection to the university and CSE dates back to 1973, because I'm an old guy. And the college was then known as the Institute of Technology. The quarter system defined the academic calendar. Classes started the last week in September, and finals ran to mid-June. So back then, today would be day 2 of week 2 of spring quarter. Now we're almost done with spring semester. So after enrolling and attending the U for a year, which was 3 quarters, summer jobs were scarce because it was the recession of 73, 75. And I didn't find a job until late July that year. That meant I couldn't save up the $850 or $900 I needed to go to school for a year, because tuition and fees were $232 a quarter, plus books. So I kept working as a machinist on the night shift. And that really worked out well for me because I didn't know exactly what I wanted to do. And I went to school part-time, studied subjects in a wide variety of fields, figured it out, and then came back to school full-time and took a degree in mechanical engineering in fall of 1983. So there was a lot of quarters between 1973 and 83, but it was the right path for me. So my wide-ranging college studies might be considered unconventional today, and I would definitely not be a favored input in the calculation of the 4-year graduation rate. But the knowledge I gained from all those varied courses has been critically important in my career trajectory. I've been in industry for over 35 years, first at Honeywell ATK, and then Tennant Company for the last 18 years. And at Tennant, we've developed many win-win partnerships with the university. These include sponsored research, internships, sponsoring projects for graduate and undergraduate experiential learning, and research center memberships. The partnerships are with a number of different colleges, including CSE, other schools and departments on both sides of the river and in St. Paul. And the U of M is truly an incredible resource for local business, along with a great source of talent. Both of the very talented, high-performing young women on my team are U of M alumni. And my current role at Tennant is Director of Sustainable Enterprise. We've got a small team, just 3 people, because our goal is to work ourselves out of a job. We just want to embed the right stuff into the existing business processes so the right things happen kind of automatically. Now that takes a long time and the playing field changes, so we keep plugging away at it. But environmental footprint reduction, sustainability, and the circular economy are longtime personal passions. And Tennant has developed many products which help our customers use significantly less water and energy and help reduce the amount of generated waste. We also work on the enterprise side, not just the product offering side. So like the University of Minnesota, Tennant is subscribed to a bunch of community solar gardens. Our market voice added over 6 megawatts of solar capacity to the local grid, while the U's voice added about 20 megawatts. On the home front, we added PV solar to our roof back in 2015. But first, we reduced our home energy usage by 55%, going back to 2007. That almost drove my wife crazy, but she hung in there with me. And that rooftop system generates about 100% of our need, plus it's half the size and about 35% of the installed cost if we hadn't done any of that energy reduction work. So tonight we're fortunate to have 3 faculty members join us whose work is intimately connected with sustainability and preserving the environment. The lecture will feature short presentations by Bill Arnold, Paul Dauenhauer, and Crystal Ng, followed by a combined question and answer session at the end of those presentations. And then following Q&A, there'll be a reception right outside the doors in the lobby area. That'll be an opportunity to speak more with the presenters and connect with other alumni, students, and friends of the college that are here tonight. So the research happening at CSE is important to the economy of the state and to the lives of Minnesotans and people beyond here. And I would personally like to challenge each of you in the audience to think about how you can implement sustainable practices into your own life. There'll definitely be some inspiration and motivation for you to do so after you hear these speakers and see their presentations. So without further ado, I'm pleased to introduce our first speaker. William Arnold is a distinguished McKnight University professor and the Joseph T. and Rose S. Ling Professor and Associate Head of the Department of Civil, Environmental, and Geoengineering. His research focuses on the fate of organic chemicals, including pharmaceuticals, pesticides, and solvents, in natural and engineered aquatic systems. He received his BS in Chemical Engineering from MIT, MS in Chemical Engineering from Yale, and PhD in Environmental Engineering from Johns Hopkins University. He then joined the University of Minnesota faculty. Please join me in welcoming Professor Bill Arnold.
So I want to thank everyone for being here this evening. I'm going to talk to you tonight about the hidden information about the chemicals we use every day in our lives that's in the bottom of the lakes of Minnesota. So I'm going to start off by saying I am not anti-chemical. Right? I recognize chemistry makes modern life possible. Pharmaceuticals, agriculture, plastics, clothing, everything we do relies on chemistry. But we have to recognize if we don't design chemicals well and use them smartly and dispose of them properly, adverse impacts can happen in the environment. So we're surrounded by chemicals. In our house, we use cleaning agents. We use pharmaceuticals. We have flame retardants in our furniture. That means they're around our bodies all the time. A lot of the chemicals we use, they go out into the environment. We put them out there on purpose, or they go down the drain. And when they're in the waters, they move around in the environment. They don't just stay in the water. They transport atmospherically. The chemicals we use here in Minnesota, we can find in the Arctic. And lastly, we have to recognize the chemicals we use are in our bodies. Right? Some of them we put there on purpose. We take medications, we put on lotions, but other chemicals come into our bodies from the environment, either from the water we drink or the food we eat. So how we look at chemicals is by looking at the sediments at the bottom of lakes. And the nice thing about sediments is they're deposited every year. That way, this gives us a way to look back in time at the chemicals that were present at different periods in history. So in the lake, there are particles. Some of them are biological, some of them are soil particles, but they're constantly raining down. And the chemicals in the lake stick to those particles. So every year, new layers of particles are deposited. And it's possible using various techniques to date exactly when the particles were put down in the lake. So we can collect a sample of the bottom of the lake by using a sediment core. So basically we take a plastic tube, punch it down into the bottom of the lake. And we do that. At the top of the sediment core is the material that was deposited most recently. And at the bottom of the sediment core was material that was deposited long ago. And using these dating techniques, the top is recent, we can date back to about 1850, which gives us a nice long profile of human history. So I'm going to tell you 3 stories. I'm going to talk about 3 different classes of chemicals. One is Triclosan, which is an antibacterial compound that was put in all sorts of consumer products. The next one is antibiotics, which you're probably all intimately familiar with. And the last is a group of disinfectants used for surfaces and as surfactants that are known as quaternary ammonium compounds, or QACs. So we've taken samples all over the state of Minnesota, from way up north to down here near the metro area. I'm going to focus on two of the lakes for most of the talk. One of them is Lake Pepin. Right, this is a widening of the Mississippi River downstream of the Twin Cities. The reason we like Lake Pepin is that its watershed integrates two-thirds of the state of Minnesota. Right? So we get a really integrated signal of the chemical pollution across the state. The other is Lake Winona, which is not in Winona, it's in Alexandria. And the reason we like Lake Winona is that at the southern tip of the lake, there's a wastewater treatment plant. And so we get a real good signal of what the humans are putting into the lake, and it's also got a very small watershed, just a few thousand feet wide. So really two end members of the groups of lakes in Minnesota. So this is triclosan and its chemical structure. Don't worry if it's bringing you chills of your organic chemistry experiences. This is just the language that chemists use to display what we're talking about. So this is a broad-spectrum antimicrobial compound. It was put into a wide variety of products throughout the 70s and 80s when we decided we didn't like germs very much. What's really interesting about this chemical is that it really got put into products because of a marketing war between liquid hand soap companies. So my product is better than your product. And it was never formally approved by the FDA for use, except for in anti-gingivitis toothpaste. It was first brought to market in 1965, which is an important date as we go forward. So we started looking at triclosan in the lab because the U.S. Geological Survey had found it in a whole bunch of rivers around the state of Minnesota, or around the country. And we wanted to know what might be happening to it in the environment. We said, well, the first thing it's going to do is it's going to go through the wastewater treatment plant. The last step in wastewater treatment is disinfection with chlorine. And we looked at the wastewater effluent, and we found that there were 3 new compounds where chlorines were added onto the triclosan ring. These are called chlorinated triclosan derivatives. So now we've got triclosan going into the environment and 3 new chemicals going into the environment that have not been tested for any sort of activity. We also found when these were exposed to sunlight, triclosan and these 3 chlorinated triclosan derivatives formed compounds known as dioxins. Dioxins are among the most toxic compounds known to humans. We've known about them for over 40 years. Now, there are 209 different dioxins. These 4 might not be the most toxic 4. But now we're potentially putting these into the environment. So now we've got a total of 7 new chemicals that are going to the environment from a compound that we use for marketing purposes. So at this point, we decided we needed to go and look out in the environment to see if we could actually find these chemicals. And you can bet that I've been talking to you about them this long because we did. So I'm going to show you a series of plots like this. The vertical axis is time. The sediment at the top is the most recent sediment. As you go down the slide, it's going back in time. And the horizontal axis is showing how much is there, the concentration. So this is the plot for triclosan. Amazingly, before triclosan was invented, there was none of it in the sediment core. And once triclosan starts being used, we see it accumulating over time. We can see the chlorinated triclosan derivatives in the core that come from the disinfection of wastewater. And we can see the 4 dioxins that form from the photolysis of triclosan, or its exposure to sunlight. And note the pattern of the dioxins looks a lot like that of triclosan, which suggests that triclosan is the source of these materials. So I said there are 209 different dioxins. There are lots of sources of them. We know about them because in the 70s we realized they were coming from incineration when we were burning garbage. And we realized that this was bad for the environment, and we changed how we do incineration so we don't make dioxins anymore. So since the 1970s, you can see these higher chlorinated dioxins with 5, 6, 7 chlorines, 8 chlorines on them have decreased dramatically over time. On the other hand, these lower chlorinated dioxins have increased over time. So over the past 40 years, we switched the source of dioxins in the environment from incineration to hand soap. So how do we deal with all these compounds being in the environment that shouldn't be there? Well, we presented this data to the Minnesota legislature, and they took action. So they passed a bill in 2014 that said as of January 1st, 2017, triclosan would be banned in all consumer products in Minnesota. And about that same time, the FDA finally decided they would do their review, and they basically told all the manufacturers they would have to pull triclosan and 20 other chemicals from their products by the end of 2017. So I'm going to change gears now and go to antibiotics. Every year in the US, we use about 18,000 tons of antibiotics. Some of that goes to people, some of that goes to animals, and that's farm animals and that's pets. Doesn't matter where it's going, it's all going into the environment eventually. So I'm going to look at two different antibiotics. One is sulfapyridine. So the sulfa drugs were some of the first brought to market in the 1940s. And so not surprisingly, that's when we see them in the sediment cores. And here the orange squares are Duluth Harbor. The purple diamonds are Lake Pepin. And the bluish-green triangles are Lake Winona. So we see a heavier signal in Lake Winona, which gets the direct wastewater impact, than in Lake Pepin, which is integrating the large watershed. If we look at a different drug, ofloxacin, it was invented in the 1980s. And so we don't see it in the sediment core until it's put into the market. So the sediment cores give us a way to look and say when chemicals are brought into the market. So we would think this would also tell us when chemicals are taken off the market. But here's when sulfapyridine came off the market. And it's still there. And so we have to start looking for other sources of this chemical. And the place we look is in our intestine. Not because it's being formed in our intestine, but we're taking another medication. This is sulfasalazine. And this is used as an arthritis medication. This part of the molecule is sulfapyridine, and the other part is essentially Tylenol. Right? So we've got a drug that's basically made up of an antibiotic and an anti-inflammatory combined, that when it goes through your gut, it gets metabolized, and the sulfapyridine is cleaved off and goes through you and into the environment. So this might not be the smartest use of a chemical. Right? We're putting an antibiotic out in the environment for treating pain. So what do we do about this? We need to start thinking about better chemical design. So this is a collaboration I'm working on with Will Pomerantz in our Department of Chemistry here. We're looking at benign design of pharmaceuticals and pesticides, particularly looking at compounds that are fluorinated. So here we have a drug that could break down into two potential products in the environment, among many, but fluoride would essentially be non-toxic, and fluoroacetate would be very toxic. Will's interested in MRI reagents, particularly compounds like this here. The one on your left would certainly be persistent in the environment. That's kind of the current gold standard. The one we're trying to design is on the right. That should be bio or photodegradable. So we're working together to design these new chemicals and then test how they degrade to make sure they form non-toxic products versus potentially toxic products when they're in the environment. Lastly, I'm going to talk about quaternary ammonium compounds, things that you certainly have in your house right now. Right? These are used as surfactants and cleaning agents. They all have this kind of same similar chemical structure. We have this nitrogen with carbon chains and a positive charge. So I'm going to compare our two lakes again, Lake Pepin and Lake Winona. And these are benzalkonium chlorides. They're in Lysol disinfecting sprays, among other compounds. And we notice the patterns here are quite different. First of all, the levels in Lake Pepin are much higher than in Lake Winona, which is reverse for every other chemical we've seen in our group. We think what we're seeing in Lake Winona is the signature of consumer use, right? This is a municipal wastewater treatment plant. In Lake Pepin, we're seeing a combination of what consumers are using as well as industrial use. And that's fortified by this large peak that we see in use that occurs in the 1970s. In the 1970s, the Clean Water Act was passed. More effective wastewater treatment went into place, as well as industrial source control of pollution. So that, we think, leads to this large drop. And then the background signature we see after that is probably a combination of consumer use and residual industrial use. So we can see here that other legislation has had an effect on our water quality as well. So these compounds are interesting because they've been in use for a long time as disinfectants, well over 80 years at this point. So one of the impacts here is on the microbiome. So this is a map of genetic material called an integron that allows bacteria to exchange genetic material. Within this integron is a resistance gene to quaternary ammonium compounds, as well as to one to mercury. And these are two of the longest kind of used antibacterial compounds. So using these compounds has changed the environmental microbiome. This integron contains these two resistance genes, and they're transferred around throughout the environment. And not only throughout the environment, but this integron is in your intestines as well. So how can we improve treatment to prevent this kind of thing from happening in the future? So this is a collaboration with my colleague Ray Hazalski in Civil and Environmental Engineering. We could use treatment wetlands, for example. The quaternary ammonium compounds and other compounds really like to stick to particles. If we could set out those particles before they reach the river, that would be a way to remove them. We could look at advanced filtration techniques for drinking water, or even adding ozonation onto our water or wastewater treatment plants to remove the chemicals so they don't get out into the environment. So lastly, I want to thank all the people who did this work. My students, Jill Kerrigan, Cale Onger, and Jeff Booth, as well as my postdoc, Sarah Patti, actually did all of these experiments and measurements. The Triclosan work was a collaboration with Chris McNeil, who was in the Department of Chemistry here and is now at ETH Zurich. Dan Engstrom at the St. Croix Watershed Research Station and the Science Museum of Minnesota has helped us collect and date all these sediment cores, and all of the analysis we did in the Masonic Cancer Center here at the University of Minnesota. And with that, I thank you for your attention.
Thank you, Professor Arnold. That was fascinating. I would now like to introduce our second speaker. Paul Dauenhauer is a professor of chemical engineering and material science at the University of Minnesota. He serves as co-director of the Catalysis Center for Energy Innovation. He received his BS in chemical engineering and chemistry from the University of Wisconsin-Madison and PhD in chemical engineering from the University of Minnesota. He's worked for the Dow Chemical Company as a senior research engineer in Midland, Michigan and Freeport, Texas. His work on catalysis and the reaction engineering of renewable feedstocks has been highlighted by numerous awards, including the DOE Early Career, NSF Career, the Rutherford Aris Excellence in Reaction Engineering Award, and the Camille Dreyfus Teacher Scholar Award. He is the co-founder of Cyronics Renewables and the inventor of the flagship technology for Activated Research Company. Please join me in welcoming Professor Dauenhauer.
Here in Minnesota, we're surrounded by an ocean of renewable carbon in the form of plants such as trees and grasses. And we call this material biomass. And we'd like to use that to create an entirely new industry of biochemicals and biomaterials. Now, why is this important? If you look at all of the things you use every day, they're made up of carbon. All these different products that you can see are made up of carbon, mostly derived from fossil fuels such as natural gas and petroleum. And so the question becomes, in 100 or 200 years, we're still going to need these types of materials in everyday use for food, for medicine, for agriculture, for transportation. And so what are we going to do that makes these types of materials and these types of industries sustainable? And to think about that, we need to go back to actually where we started and what we do today for the most part. And that's the status quo of the carbon chain. So right now, we take carbon out of the ground in the form of natural gas or petroleum. Out of that, we pull out chemicals such as 2, 3, 4, 5, or 6 carbon chemicals, like you can see there. And then what we do polymerize, where we connect those into big, long chains. And those are heavy, and that's what we think of as solids. We call those plastics, or the chemists call them polymers. And ultimately, then, we can put those into the products we use every day, like bottles or shirts or cars. Now, the problem with this is multifold, right? The feedstock here is non-renewable, right? There's a limited supply available on Earth. And on top of that, there's waste generation. What happens at the end of life of these types of products, polymers and plastics? Well, what we can do is think about improving that entire system by looking at this several different ways. Number one, we don't have to take the carbon from fossil fuels. Here in Minnesota and many other places, we're surrounded by carbon that comes out of CO2 from the air and is stored as biomass in trees, grasses, and these types of materials. And the other thing is, it's not just a one-way path. We take the carbon from biomass, we make chemicals, then we make plastics, then we make materials, but we can take this back the other way. We can recover the polymers and plastics from materials, recover the chemicals and use them again, or we can biodegrade something all the way back to biomass again. So the question is, how do we do this here in Minnesota? In Minnesota, if you look at a map of the United States, it identifies, this was put together by the US government, lignocellulosic biomass. This is the types of non-food biomass that exist around the country. Minnesota is strategically placed in the sense that we have access to a lot of carbon that is renewable. And that comes in the form of forestry in northern Minnesota, grasses, and agricultural residues like corn stover in the south. So the question is, if we were going to put biorefineries around Minnesota to process that carbon to the materials we use every day, how are we going to do that? And that goes back not only to chemistry and engineering, but also economics. We have to be competitive with fossil fuels if we're going to replace them as the feedstock of choice. And so we can think about where we start with. Well, from all these different biomass and plant sources, we pull out a molecule called glucose. And you can see it here in green. It's this hexagonal shape. And from that, you can look at the price. The cost to take that out is about 10 to 15 cents per pound of carbon, which is actually quite low. And if you think about where we're going, if you look on the right, this is one example. This is a molecule on the right in red called paraxylene. And that's the key molecule that shows up in polyesters, such as plastic bottles, clothing, car parts, etc. And that's actually a relatively cheap chemical. It's about 60 to 80 cents, depending on when you buy it and where you buy it. So we have to think about a process that can take glucose in green that we get from biomass and convert it to paraxylene cost effectively. And there's several ways to do that in terms of chemistry. In this picture, all of the dots are different chemicals, and all the lines are different chemical pathways. So of the hundreds of options, we can think of kind of a couple of these. If you look at the top path, you can see that you actually end up with a price above commercial paraxylene, which means it's too expensive, and we wouldn't be able to make that cost effective. Whereas the other processes actually end up below, and we can think of those as cost-effective technologies. So what are the technologies that actually make this economically viable? Well, this is what we work at here at the University of Minnesota. What are the types of chemistries and process technologies that can do this? And here's one example we've been working on for the past decade. On the left, again, we start from glucose, which is what we can get from grasses and trees. And then we can convert that 6-atom ring, that hexagon, to a 5-atom ring, which we call furan, and then convert that even further by taking off the oxygens. Now, the key contribution to this, the part that hasn't been known for a long time, we discovered here at the University of Minnesota, where we can actually add another molecule that we get from ethanol, and then we can, in 2 steps in the reaction you see here, we make this molecule paraxylene. Now, the key part of this is that we use what's called a catalyst. A catalyst is a material which can accelerate the chemistry and make the reactions do what we want, essentially. And they're difficult in the sense that we have to make these atoms behave the way we want. And so we start out, usually they're not very good. You can see this in the plot on the left. What you're looking at here is essentially the performance in pink, the yield of the molecule we're trying to make. And you can see when we started out, we were getting very low yields of about 30 to 40%. And over time, as we keep improving these catalysts, we eventually get up to something that's over 97% effective at producing paraxylene. Now, what is that material? Now, what you're looking at here in this picture is an electron microscope image in the top left, where you're actually looking at nanoparticles. And these particles are about 200 nanometers in size. So if you lined up 200 of these end to end, they would be the width of a human hair. But within these are actually what we call nanospaces, or pores, where molecules can go in and react and do the chemistry we want. So we can design those holes, those nanopores within the structure, to direct the chemistry the way we want. And the effect of this, being so efficient at making the molecules we want, has a huge benefit for the economics. So if you look here on the right, this is our progress in terms of performance of the catalyst each year compared to its impact on the cost of the biorenewable chemical. And what you can see is that when we start, the catalysts aren't very good and the price is high. And as we improve and improve, we eventually cross over the red bar, which is the cost to make to sell paraxylene made from fossil sources. So this is now an economically competitive biorenewable chemical that goes into a lot of products that we use every day. And there's lots of chemicals like this. If I go to another technology, again, we can start with glucose that you see on the left that we can get from plants. And in 3 steps now, I can go from glucose all the way to the molecule you see in the top right, just to the right of that red arrow, called isoprene. And if you take isoprene and link many of them together, you get a big polymer, plastic, that we call polycis isoprene, which is essentially a car tire. Now, how do we get there cost effectively? Well, we want to do this in as few steps as possible, and we want the catalyst to be as efficient at each step as possible. So what you can see here is in the second step, we use a nanoparticle made up of two different metal atoms. And we can tune the composition of those nanoparticles to be as efficient as possible in that chemistry. So you can actually see if I change the amount of rhenium versus the amount of palladium, at about 20% rhenium in that nanoparticle, I get the maximum rate of conversion of that chemistry. And then if I take the catalyst we invented last time, the nanoporous material and incorporate that into the third chemistry, I get an overall highly efficient process to go from glucose from plants to the key chemical in car tires. Now, that's just in the laboratory, in little beakers and stirred dishes. But to go to something like a big refinery, we have to scale this all up. And so when we do that on paper, it looks like what you see here in this picture, and there's a lot of details you don't need to know, but we can cost out how much does it cost in these big containers and how much it costs to operate. And when we do that, based on this design, we can actually estimate the cost to make renewable car tires. And so what she's looking at here in the kind of purple bars is the price that we could manufacture renewable isoprene right now. And the colors you see in blue are actually the prices in India and the United States for isoprene on the market. Now, we just started this. It's only about 2 years old, and we're already close within striking distance of making renewable car tires here in Minnesota. But you can see we've identified 3 significant advantages to the catalyst that we think are realizable in the next 5 years that can make this economically competitive. Now, this is a very unique way to go about this. We're going to make the exact same molecules that we get from petroleum, and we're going to substitute those into the products you already use. So we call these drop-in replacements. And there's all the molecules that are made from petroleum we can try to make from biomass, which is a good strategy because then we don't have to redesign all the products you use. The other approach, though, is that biomass has a different structure, which means we could make new molecules which have new functionality and actually behave better in different applications. And my third story, I'm going to talk about the molecules on the bottom. That has to do with laundry detergents, which we know from the previous talk, laundry detergents are very important because of the number of chemicals we use. And so what we could do is make the molecule you see in the bottom right. That's called linear alkyl benzene sulfonate. That's the key ingredient you use when you wash your clothes with detergent. And we're going to propose we use the molecule on the bottom left, which we can make from biomass. Now, if you think about a formulated product that you buy, and you read off the list of chemicals in that, there's a lot of things in there, but it has to have the active ingredient, which in this case, in blue there, is linear alkyl benzene sulfonate, which we get from petroleum. And we put a lot of that in there to make it work. We also have a lot of water. And then everything you see here in green, orange, red are added chemicals that have to go into that formulation to make it work in things like hard or cold water. And we got looking at this, and we said, well, what would make the economics really good for this is to not have to add all of those extra chemicals. Let's make an active ingredient that you see here in red, which we call oleofuran sulfonate surfactant, that can actually act and clean your clothes without all of these other chemicals in your particular system. Now, the molecule that we invented here has the same functionality as the conventional chemical, but it behaves better. For one thing, it's tunable. If you look at the structures on the left, right, you see that it has similar functionality to the molecule we're looking at, but now we can change little parts of it. That means we can do things like change how foamy it is. If you want to wash your hands with a foamy soap, we can tune that into the molecule. If you want to put detergent into your dishwasher and you don't want any foam, presumably, right, we can take that out. And so that's a unique capability, which means we can put these molecules right where we need them. But the other capability, which only shows up in this biomass-derived molecule, is the stability. So if you take any other surfactant and you use it in cold water or use it in hard water, right, it actually stops functioning. It'll form crystals. And you can see that in this picture you see here on the right. So if I take a vial, like you see here on the right, and I put behind it the Minnesota logo so you can tell if it's clear or not, I can actually put the surfactants in with different amounts of hard water. So you see the LAS is the surfactant, the active ingredient in most laundry detergents. And by the time you get to 230 parts per million, which is where common hard water is, it actually starts forming crystals and stops functioning. But the OFS, the biorenewable surfactant, is crystal clear even up to 10,000 ppm, which is like seawater, which means we can use these molecules in all sorts of places without having to add all of these other chemicals. Now, the ability of the stability combined with the ability to tune these molecules means we can use them all sorts of places. And here at the University of Minnesota, we've been thinking of all the different application spaces. And we patent the technology so that we have the ability to translate it out into the marketplace. And so if you think about these molecules, we tune them. Then when you put them in water, they form these spherical structures. Those structures can then be tuned to give all sorts of different applications. So you can use these in paints. You can use them in detergents, of course, but other places that you wouldn't think of too, such as foods or agrochemicals, where you're going to put this in and spray pesticides or herbicides on plants. Now, this technology is being translated to the marketplace by a startup company out of the University of Minnesota, which is Ceronics Renewables. And that actually is led by a PhD student here from the University of Minnesota, Christoph Krumm, and their flagship product is actually one of the molecules I've just shown you on the previous slide. Now, what's exciting is in the next 2 decades, we have the ability to completely remake the chemical industry here in the state of Minnesota. Like I said, we're uniquely placed in terms of feedstock, but we also have, right, the background agricultural setting and the people and the infrastructure to move this carbon around and manufacture all of these products. And now we know what's been a problem for a long time is that people think these materials can't compete economically with petroleum. And actually, it's the reverse. Some of these technologies, when they get implemented, such as technologies like renewable lactic acid, already compete and outperform fossil-derived chemicals. Now, on top of that, we can make new chemicals, right? The chemicals we make right now don't have to be the only ones we use forever. With biomass, it has new functionality, and we can outperform everything else. And like I said right at the beginning, Minnesota is a unique place for this. We have the feedstock, right? We have the agricultural background, and the University of Minnesota has a lot of expertise in chemistry, engineering, economics, and technology translation. So thank you very much.
Thank you, Professor Dellenhauer. Finally, it's my pleasure to introduce our third speaker. Crystal Ng is an assistant professor of hydrology in the Department of Earth Sciences. She received her bachelor's degree in applied mathematics from Harvard University and PhD in civil and environmental engineering from MIT. She worked at the U.S. Geological Survey before coming to the University of Minnesota in 2014. Crystal is interested in how the movement of water in the ground impacts vital resources in our environment. She focuses on how water contaminants migrate and geochemically evolve, as well as how vegetation grows in response to water distributed throughout the soil. She'll be talking tonight about research on wild rice, which combines her interest in water contaminants, ecology, and natural resource protection. Soon after she arrived in Minnesota, she began researching the fate and transport of sulfate on the Iron Range in northeast Minnesota, which poses a threat to wild rice. This work has grown into a collaborative and interdisciplinary effort that examines both the biophysical and human dimensions of wild rice. To this broader project, she contributes expertise in the flow of water between groundwater systems and lakes and streams, contaminants in wetland, stream, and lake sediments, interactions between water and vegetation, and field and computer modeling methods. Please join me in welcoming Professor Ng.
Wild rice. It's Minnesota's state grain, and I'll bet it means something special to a lot of you here today. Does anyone make wild rice for special occasions? Yeah? Okay, right. Who has a favorite wild rice recipe? Maybe a family recipe? Yeah, some of you here. Who has been out ricing before? Okay, so I planted a few people in front of the room here. But okay, so for those of you who don't know as much about wild rice, a lot of what's sold in stores is actually cultivated or farmed wild rice. The really wild rice, that grows naturally in shallow lakes and streams across the Great Lakes region, such as this one here. And you can see from the other picture, they really are different. Natural wild rice has this nice varied coloring, and its taste and its texture is quite distinct. And you can buy natural wild rice in stores. Next time you're shopping, just check the labels. Unfortunately, though, natural wild rice is highly sensitive to a multitude of environmental stressors. This includes fragmented habitats and high water levels, and it includes warming temperatures, interestingly, warming winter temperatures. Wild rice is vulnerable to different invasive and competitive species, and it's highly sensitive to water contaminants, such as sulfate, when it transforms into the toxic form of sulfide. And all this has led to the near decimation of wild rice in Michigan, as well as the impairment of many wild rice lakes and streams across Minnesota and Wisconsin. And when there's a problem for wild rice, there are wider implications throughout the ecosystem. Many waterfowl depend on wild rice for their food or for their breeding grounds. And so you can see that this sensitivity of our beloved state grain really is cause for concern. In fact, it can be devastating for Native Americans in the Great Lakes region. For them, wild rice is not only an important part of their diet, but it is central to their cultural identity. For the Anishinaabe, or the Ojibwe people, Their migration story revolves around wild rice. Their creator told them to go to the place where food grows on water. And so wild rice has been it's been a sacred guiding food for them, for where they are today. And today, tribes continue to harvest and prepare wild rice, and wild rice plays an important role in their ceremonies. And today, tribes are sovereign nations whose access to wild rice is legally protected by treaties signed by the US federal government. So for all these reasons, wild rice really serves as a flagship for both environmental preservation and for indigenous resource sovereignty. Of course, for the same reasons, wild rice has also served as a flashpoint between tribes, industries that affect wild rice waters, and state agencies that regulate them. And this problem captivated me from the first time I ever heard about it when I arrived to Minnesota 5 years ago. I'm a hydrologist interested in how the movement of water and contaminants and the growth of plants interact with each other in ways that impact society. And so I began researching how the exchange between groundwater and surface water can affect the levels of sulfate and sulfide that accumulate around wild rice roots. But I began to realize that all my hydrological and geochemical computer models, they couldn't possibly tackle the full complexity of the problem, especially not when it comes to those sensitive cultural and policy issues. And so a couple years ago, I assembled an interdisciplinary team from across the university to examine both the ecology and the policy of wild rice, all under the consideration of its cultural importance to Native people. And we received a grant from the university's Grand Challenges program and Institute on the Environment, and our aim has been to support tribes in their work to protect wild rice. So, we're about 1.5 years into the project right now, so you're probably expecting that I'm going to show you a bunch of plots and data from all these different wild rice lakes and streams that we visited, right? Well, actually, this slide showing our full project team with all of our students and all of our tribal collaborators. This slide is actually what best sums up our greatest achievements thus far. So let me explain. We started off the project with 6 letters of support from different bands and tribal organizations. So I've got to say, we felt like we were all set with our tribal partnerships. And so when we got our grant, we were ready to dive right into our research. But we could feel most of our tribal collaborators really hanging back. You know, this really confused us because hadn't they written us these letters of support? Well, they pointed out something to us that's going to sound really obvious to you right now, but it's something that we, as researchers, too often forget when we are just so fixated on getting that data and getting those publications out. And that is, a true collaboration takes much more than just securing that letter of support. A true collaboration is a relationship among individuals, and it takes time, goodwill, and a lot of listening to develop. And this is especially the case when working with communities that have been repeatedly marginalized and have had their concerns ignored again and again. And so we did something that I certainly have never done before. We put aside our research plans. And instead, last spring, we traveled around Minnesota and Wisconsin, and we visited different reservations and tribal organizations. And we listened. We listened to what tribes were concerned about with wild rice. And we recruited a group of really bright Native and non-Native students, and I'm glad to see a few of them here tonight. They are proving to be invaluable bridges between our research team and our tribal partners. So what we have found is that with the very simple act of us listening, many tribes have been willing to do something that is not so simple, and that is to engage with us, even after so many negative experiences that they've had with researchers. And so that's why today, right now, I am honored to show you this list of individuals, which includes representatives from 11 different bands and tribal organizations, all of whom expressly wanted to be named as a collaborator on our project. And there are many more beyond this list of people who have been willing to share their perspectives with us and help us really understand why they have so much to be wary about. And our project was given this Ojibwe name, Kawe Gida Na Na Gado Ndam Manoomin, to remind everyone on the project of its guiding principles, that first, we must consider Manoomin, or Psin in Dakota, or wild rice, because wild rice is profoundly important as both a natural and cultural resource for Native people. So just 1 year ago from right now, we had no idea of any tribes would want to work with us. But since then, we've been coming together to the table to hatch new research plans, and we've been going out together to learn about wild rice lakes and streams. But I do have to admit that establishing and maintaining a trusting partnership takes a lot of time, and it can be really hard work. But all of you already know this from all the important relationships that you have in your lives, right? And so you also know that all that effort is absolutely worth it. In our project, so far, we only have preliminary results, but it is already clear that a close collaboration with tribes is making our science so much better. You see, we started off with only 3 study sites in our original research plans. All were in Northeast Minnesota, and they were chosen based on their levels of sulfate. Well, our tribal partners helped us realize that our thinking was too narrow. It was far too narrow if we actually wanted to figure out what the problem with wild rice, because it's probably a combination of all the stressors that I told you about before. But the stressors are different across the Great Lakes region. And so tribal resource managers from 10 different bands and tribal organizations helped us expand our view to include over 30 sites that they were concerned about. And these span a far more representative range of the hydrology, ecology, and management conditions out there. And our tribal partners, Eric Chapman and Joe Gravine from Lac du Flambeau, they put it best, I think. And we're actually fortunate to have them here with us tonight. So Eric is hi Eric. Eric is tribal council member and the program director of the Wild Rice Cultural Enhancement Program. And Joe Gravine is a wild rice technician who is passionate about protecting wild rice for his people. They explained to us that Manoomin, or wild rice, has no state or county boundaries on where it will grow and provide sustenance for our families and a healthy environment for the winged ones, the ones that swim, the ones that crawl, and the 4-legged ones. We recognize Manoomin as an indicator of healthy Nibi, water, and hope this project educates our future generations on the importance of this gift from the Creator. So last summer, we were able to make it out to 9 of our tribal partners' sites of concerns, 5 of which were located across 4 different reservations. And they included everything from threatened lakes like this one, where perennial competitive plants were just starting to appear, as well as sites that were already impaired, such as this stream reach that once had productive wild rice stands, but no longer does not. Now, I do have to pause here and make a very strong cautionary note. You might be thinking so far that the main advantage of working with tribes is site access. But that perspective of site access is fraught with colonial history. And more specifically, it has caused confrontations between researchers and tribes. In our project, what has been truly invaluable is not unilateral benefits of site access, but it's been the exchange of ideas, and it's been those perspectives and insights that our tribal partners have been willing to share with us. They've been sharing with us the histories of these sites, and they've been helping us find the best places to be collecting samples, all of which would have been just hugely challenging for us to do at so many different sites. But our tribal partners aren't simply introducing us to these sites, they're also intellectually driving the science there. Our tribal partners have come up with so many fascinating hypotheses that we never thought of. And these come in part from their close familiarity with their own lakes and streams. But it also stems from their cultural way of thinking about the environment altogether. And this is in contrast with our tendency in academia to isolate different components into different specialized disciplines, which I'm beginning to realize can make us really myopic in our interpretations. For example, as a hydrologist, I usually only think about the movement of water as bringing in those contaminants that we find in lake beds. Well, some of our tribal partners asked whether airborne dust that settles into lakes could be another mechanism, especially in this one lake where we really haven't been able to figure out a hydrologic source. Here's another example. Early on, we drew a bounding box tightly around the aquatic system, and we figured we just had to resolve those different arrows inside. Well, a couple of our tribal partners asked whether we actually made sure we drew the box in the right place. They pointed out that at some of their lakes, the surrounding areas have been intensively logged, or they're highly vulnerable to changes in tree cover. And they noted to us that changes in upland vegetation could be changing the lake levels nearby, or they might be changing the amount of runoff of nutrients into them, both of which could negatively impact wild rice. So I can tell you that there's so many new hypotheses that our tribal partners have shared with us, but I'll give you just one more. Okay, so quite frankly, our team has struggled with stepping out of our comfort zones to think about things outside of our areas of expertise. And this has been an obstacle for doing interdisciplinary research that actually integrates biophysical science and social science. When we do try to stop talking with each other, we often end up really fumbling with each other's jargon. So I was really scratching my head with this one term. Okay, I'm looking at May here. Okay, cultural epistemological frameworks, something like that. Okay, right? And at the same time, I was trying to explain this concept of chemical end members, even though to me it was like the most obvious thing in the world. So, okay, so here we are really struggling, right? But one of our tribal partners stepped right in and said, look, I'm really concerned about losing traditional harvesting practices and whether this could have a negative impact on our wild rice. So what he explained to us is that his elders always taught him to pluck out competitive plants during his harvesting breaks, but he's noticed that youth don't always learn to do this anymore. And he wondered whether this could be contributing to the worsening problem of competitive plants crowding out wild rice. And he wanted to think about how he could get youth in his tribe to attend ricing camps like this so that they can learn these important lessons that could help preserve their wild rice stands. So clearly, our tribal partners have no problem seeing these potential links between biophysical science and social science. So we really have a lot to learn from them about interdisciplinary science. Okay, so all these ideas that I've given you so far, they're still just hypotheses. We don't have the answers yet. And that's because the essential first step has been to establish a strong and trusting relationship with tribes. But one thing we do now know is that wild rice is profoundly important to Native people. And when we, as researchers, can understand this, then we can begin to learn about the diversity and the complexity of these lakes and streams that have been part of their lives for generations. And we might understand aspects of the environment that we wouldn't have been able to figure out so easily on our own if we just use our usual academic approaches, which can be too often too narrow. So I'd like to end by noting that all these examples that I just gave you for how working with tribes really improves our science, they've all focused on the intellectual aspects of our work. But what does make good science? Is good science solely those innovative discoveries about physical mechanisms and chemical and biological processes? Well, I would argue that the definition of good science is more than that. I would argue that good science is also impactful science. It's findings that improve life conditions, equity, and the sense of identity within communities. So, how do we do impactful science? Well, I'd like to thank our tribal collaborators for helping us figure this out, because I know it's going to be one of the most important contributions of this project. And we are privileged today to have a few of our close tribal collaborators here tonight. As I mentioned already, we have Eric Chapman and Joe Gravine from Lac du Flambeau, and we also have Karen Diver here, former chairwoman of Fond du Lac. And so I'd like to thank them, as well as all of our tribal collaborators, for teaching us that when we want to do research for people, we must do research with people. We must really listen in order to understand what is truly important to them, and to know what we must consider to make sure that we are doing our research in the most positively impactful, respectful, and meaningful way. So, miigwech. Thank you.
Thank you, Professor Ng. I'd now like to welcome all three of our presenters back to the stage for a brief Q&A. And so the audience should, you saw a lot of fascinating work. I know you have questions, so start thinking about your questions, and there'll be a couple folks that will come and find you if you raise your hand. But I'll start with one question, which is, when I listen to all of you describing your work, collaboration and cross-functional, interdisciplinary is a big part of it. Could you just talk a little bit more about how you develop those relationships with other department members and researchers?
I think that's what my entire presentation was about. So maybe I'll let them explain, because it's been the central aspect of our work.
That's a good question. I mean, part of it is coming to events like this. As Paul was talking, I was thinking, oh, I have a project that Paul could plug into. And being aware of what else is going on in the university is hard. It's a big place. But trying to make sure you're out there and engaging with other people is a big part of it.
Yeah, I think for what we do, it's very unconventional. And we have upstream partners that do agriculture, and we have downstream partners that do product development. And that's all things that people work on in the university. But it's more than that, too. It's economics, process systems, even civil engineering and water, right, is affected by chemicals that we manufacture. And so we're always talking to people and finding partners to understand our problems.
So it sounds like never underestimate the power of the network.
I'd say yes.
So do we have any questions from the audience? If you do have a question, raise your hand.
So I'm glad you two are sitting next to each other because, in our rush to innovation and, we've ended up with problems later, right? Because we didn't take the time to really look at multiple impacts. And, I was Googling some of the compounds that you were using to make your palladium. It's popular where copper nickel mining is done, which is very water intensive, very water impactful, toxic, could be toxic, liver and kidney issues. And so, it's hard not to, in trying to fix one problem, to create a set of new problems. So how do we make sure ethically that we are taking the diligence and not the rush to market? Because too often I think we're driven by the financial rewards and not by the impact on good science and impact on humans and environment.
Hi, I guess I can give an answer. As a co-founder of a startup company, I wish there was more of a rush to market. It takes a very long time to get a product to get out there. And I'll say that the one thing we look at a lot is if we make a new compound, what are the impacts of it? So we actually do testing like biodegradation and toxicity testing within the startup company. So it's not that we make a compound and we only look at the economics. There are many other factors, both performance and environmental impact, and that's all part of the development process. So across a slate of chemicals, it has to pass all the tests, including any government restrictions for what we're doing. And that's what takes so long to bring something to market now. There's too much risk to just put a chemical out that isn't known. So I think we're pretty cognizant of that, especially now, and where the chemicals are used, if they're used in consumer products, where they'll show up in the human body or in the environment.
And I'll just add that I don't think companies are trying to do things to put profit over planet. Some certainly are. Many do not, right? They are going through all these tests. There's always going to be something that we don't anticipate, right? And so it might be a reaction with light or another interaction, and it's going to be a challenge to find that in the testings that's mandated. And shoot, I had another point. Maybe it'll come back to me.
I think.
I remembered. Sorry.
Good.
We're never going to have zero risk, right? Humans are constantly altering the environment, and everything we do is going to change something. And so we can't have a modern lifestyle and zero environmental risk, and we're going to have to live with that to some extent.
I do think that companies are getting a little bit better about managing risks long term, and there's a lot of bad examples out there, but many people have figured out that you really only have one reputation, and that's pretty important to maintain and preserve. Any other questions from the audience?
Hi. I believe Dr. Ng mentioned something about academic myopia, the notion of if you're an academic, you understand, or you think about, maybe one mechanism at a time. And I was a physics major, so I definitely understand that. What opportunities do you see for evading that sort of traditional myopia in your work, all three panelists, or what barriers are you still trying to overcome?
Well, I think, I hope I've really impressed upon everybody that really learning from our tribal partners and seeing their example of how they think across lines has been invaluable for us. And, we're hoping that they brought up so many things that we hadn't thought about before. And so they're already getting us talking to each other, like those of us in our different isolated disciplines. And so I think we really just need to listen to each other and think about ways to actually make those connections and not get so caught up in just, our one very narrow area of expertise.
Yeah, my students work in a laboratory, so it's a nice laboratory, but it's inside all day, 5 days a week, and so I think for them it's important to go out and talk to people that are doing something different but is peripheral. And I can tell you an example of this. The university here runs the MinCore program, which is an NSF-funded program to teach engineers and scientists how to go out and speak with business people and talk with customers and understand how their research and technologies interface with the industrial and economic world. And I can see that has a big impact on the students. So getting away from myopia of what you're doing involves forcing yourself to get out and talk to people that are connected to your work but don't do anything like you do, and you'll hear new things. It's also re-energizing to hear something different about the area you're working in, and it just makes you do it more, I think.
Yeah, and I'll just comment that I think, I'll reiterate, listening is very important and something that's hard for us to do sometimes, right? Because we're so driven to do what we're told to do. And I think the reward structure that we have for both how students are engaged and how they're rewarded in faculty has to be taken a look at as well.
So this is specifically for Professor Dauernhauer and Professor Arnold. Professor Ng, feel free to comment if you have anything. But the map that you showed of the National Renewable Energy Laboratory's estimate of biomass resources doesn't necessarily account for lands that are that indigenous people live on or that are used for really important resources. And so biomass utilization has important implications for like the food versus fuel debate of we don't even have necessarily enough food to feed everyone in our country or the world. So how can we start using these resources for other products? So in what ways do you see the industry and academic research trying to address those kinds of issues?
I think that's a great issue. I get this question almost every talk I give. If we're going to take carbon from biomass, how is that going to affect everything around it? Can we take it from that place? That's multifaceted, and there's people that do research in all of these. For example, take the sourcing of carbon from the environment. Right? There's food biomass like corn and soybeans and many other types of food type crops. And then there's things like wood and grass, which we're not going to eat and we can't eat. And these can grow in places where you wouldn't grow corn and soybeans. So the figuring out where these places are, where you could site biorefineries that would take advantage of this is actually a research area that we do in our department. I think, what was the other part of your question? The sourcing and also the...
And the rights of indigenous peoples on those lands.
Right, so I think going back to the sourcing and siting, right, obviously wherever you would implement something like this, that's all going to play into who owns the land and what's permitted and the effect on the environment. So obviously you're not going to clear-cut a forest for these types of feedstocks. There's industrial forests or grasslands and more ecologically beneficial types of methods of growing grasses and trees that are good for these types of applications. So, and those have to be worked out every place you'd work, just like you would with any other agricultural application. But it has to be done in an environmentally responsible way.
I'll just comment, certainly if you start changing what you grow, you're going to change your water footprint as well. Right. You have to think about that in all this too.
So I was just curious when I saw the different variations of soap molecules that you were developing, and you said that you could you could alter them to make the soap more foamy and such. How exactly do you do that? Because I didn't really see that detailed in there. I'm just curious.
We can talk for hours on this, I'm sure. So I showed a picture where the molecules come together. That's called self-assembly, and they form things called micelles. And if you change the structure, they fit together a little bit differently, and then that affects the surface tension. And where you change the surface tension changes whether you make bubbles that are stable or bubbles that aren't stable. And so one thing I've learned, just because I talk to a lot of people that make hand soaps, is people want foamy hand soap, which apparently doesn't do any good, but it's your expectation, right? I didn't realize that going into this, but I've known, actually, that you can't put these types of things in a dishwasher. So how you engineer that in is actually quite complicated, and some of it's trial and error, and some of it is a more elegant approach. But it has to do with how the molecules fit together into that little micelle, and if they're more branched, they don't squeeze together very well.
Okay, cool. Thank you.
Yep.
It's going to take a big cultural change to decouple soap and bubbles. So we've got time for one more question. I hope we've got someone in the audience that can pose something. Thank you.
Thank you all for your presentations. I have a question about the biomass plastics and wanted to learn a little bit more about the end-of-life cycle considerations for this. So for example, with plastics and rubber derived from biomass, is there more potential to break down these plastics at the end of life cycle,
Yep.
More so than fossil fuel-derived plastics? I'm thinking particularly of problems in the environment like microplastics and how long plastic persists in the environment. Thank you.
Yeah, this is a huge issue, and I'm sure it touches on water a lot as well. So I showed a picture where I said sustainability has to be more than just biomass sourcing. And here at the University of Minnesota, we have the Center for Sustainable Polymers, which, if you look at their mission statement, which is long but comprehensive, it says we want to source things renewably, but then we also have to think about the end of life for the polymer. So this is several things you're talking about. So some of the polymers we use now, we can add things to them to make them recyclable, right? Or we can add things that make them break down, right? Other polymers, we can add in functionality that make them biodegrade, right? There's many different approaches for many different applications, but it's something that we have to think about. How can you recycle these? Some of the polymers, right, you actually have to break all the way back down to the chemicals and rebuild back up again. Others, we can add new technology to them to make them recyclable and reusable. But for every application space, we're developing these technologies right now. In fact, just a block from here, a lot of these are being developed. I don't know if you want to add. There's a big impact
I'll just
on
add,
water.
as part of this design too, and especially the microplastics, the plastics carry other chemicals with them. Right? A lot of the chemicals I showed, they'll stick into plastics and then get transported other ways. And so if we can design the plastics better, we could potentially prevent that from happening. Or if they degrade more rapidly, they don't have the chance to take these other chemicals over long distances.
Well, I want to thank you all for coming, and we hope to see you at a future Curiosity Drives Progress lecture. Please join us for light refreshments outside the theater doors in the lobby, and thanks again.